We're developing a regenerative medicine solution for circumcised men.
So at the moment, there's multiple samples there. The bioreactor is basically just a huge glass blast with a lot of bells and whistles that are helping to either grow cells, or the one for decellularization, there's a bioreactor that actually helps the cells to really go away from the structure. So this is a piece of art. The clean rooms are behind me, so all the air filtration we can see and all the machinery around us is keeping the air pure without germs and at a precise temperature and precise air pressure. We have different types of rooms in here. One is dedicated to bioprinting, or our bioreactor system at the moment is clean, ready for the machinery to arrive. And then the second one we're gonna see is full of the equipment at the moment. The decellularization is running there. The bioreactor is in a ready to go stage, so we are doing some maintenance on it and taking it. And also there are some other samples in the processing. So we're gonna get through suit and we're gonna go inside. So as we are going to enter the clean rooms, we need to change and take the overalls that protect the clean rooms and all the samples that we brought with us. I'm not wearing a hairnet for obvious reasons. So, covering shoes, covering myself, that's the name. So, face mask. So at the moment we're in a clean room where there is controlled air pressure, no germs, and here we can work with the samples. So when the samples get here and they are crossed, you can start to manipulate them in a sterile environment. So we prepare everything till here, so everything is ready, sterile, and then we can go from this station to the bioreactor itself. There are multiple strategies and multiple protocols for which you work, but the absolute priority is sterility, and always work with the time windows so we don't forget from the sterility. So that's why we are so covered up. So this is a small volume 5 reactor. Just for testing out the systems, everything is sealed and the pressure is okay. So basically we need to, depends if we are decellularizing in 3D or in 2D. 3D meaning the foreskin is a cylindrical shape, so it's a cylinder. Or a 2D, there's a surgeon is going to cut the foreskin and the process is going to open, usually the butterfly, the pigs. So either we're going to set it up for 2D or 3D, we need to attach it to the system inside. Because the foreskin doesn't need to just flow around, there is a specific amount of RPM and everything, so we are trying to control it. For the 2D, what we use basically a high quality stainless steel mesh that is actually quite flexible, and it allows all the reactants and all the solutions pass through to the tissue and through the tissue and again out. So this is basically like a membrane that helps us all the tissue and is also non-reactive. That's really important for us. The thing is, there is a high quality stainless steel mesh. You need to cut it, press a varnishing, prepare some mounting holes, and also there's an option you can coat it with the blast around just to avoid the sharp edges. So what it is, a raw form looks like this. This one's prepared to be coated and then attached with a tissue to the bioreactor itself. For the attachment, as I mentioned, there are multiple approaches, either through the main port of the main head or through the side head. So depends again if we are doing it 3D or 2D, but the major stays that the main idea is to pump solutions in and pump some solutions out. So it depends on how many solutions you have. You can have a lot of connections. They're actually pretty easy to connect. So there's just the simple connections, but yes, it's sterile. So every time you open it, close it, it's sterilizing. So every time all these connections need to be sterilized. All of it's actually stainless steel, high quality, so you can either put it in an autoclave or you can sterilize it via UV or some ethanol solution. So depends. Then everything is then hooked with the sample, and it's just inserted inside the bioreactor, sealed, and then moved to the bioreactor chamber that's over there. So, okay, basically we have also some different solutions that we use, some different tools that we use. for tweezers if we need to anything. Everything, of course, kept sterile. There is a bag of sterile bags, so we have built everything ready. We need to seal it in the sterile bags, so that will be sterile. We have some measuring tools, so we know the diameter of the sample. So that's also very important for us. And everything still needs to be, there's another disinfectant inside, so every time we're working, it's almost still repetition is target is number one priority. As all of this is going to clinical research and technical outcomes, we really have to be really strict and trying to push the highest norms. So we can basically avoid these norms, more trying just to be like, this is just research. We can just play around. No, we are doing clinical research, so there's absolutely no room for mistakes or for any non-sterile equipment. When the samples are ready, what I can show you, so this one is actually sealed. So let's check the 3D decellularized foreskin that has been sealed, and it has been sealed yesterday. So now we can see a 3D decellularized foreskin. This is the cylindrical shape. So that's completely foreskin. The procedure, the surgeon was really cutting the foreskin in here. You can see precise the protocol. So now you can see it's a bit foggy because it's cold. So you can see the decellularized foreskin. This decellularized foreskin then is either tested or tested with the badge, depends. Which if it was a whole badge, this one. So we can take a small sample and test it. And then it can go for reseeding, recellularization, further processing. Many of these actually end up in analysis, so we need to analyze it. Biomechanically, histology, immunohistology, if there's a residue of human DNA, bacterial DNA, fungal DNA. So we are doing an insane amount of tests in certified laboratories that are connected to us, because we are capable of only a certain amount of tests. And the quality, as mentioned again, we are still pointing for clinical research and the clinical outcomes, so we really need to use some laboratories. They are certified for this type of test. So there is no way around. And as mentioned, it's still in sterile solution. There is a gas still inside, so it's tight, but it was still sealed with sterile environment. So when they go out, now I can manipulate with them, but they are still sealed sterile. Then when this foreskin is going to be used, it can be stored for some days here. But you have to change the solution, and you really need to take care about it. Or you can froze it, as we spoke at the beginning. The samples can be pre-cooled, so they are pre-cooled. Then in a sterile environment, put on a sterile piece of aluminum. Due to the thermal regulation, it's a good thermal conductor, so it could really shock freeze. It's placed on a piece of aluminum and sealed in a sterile bag, and then it can be shock freeze in -80. If we want to store it further, then we can dip it in liquid nitrogen. So it depends. So these are the decellularized sample, the huge one. And for some experiments that we are doing with the decellularization, so the first experiments that we are trying to find out, there are small 10 millimeter in diameter cuts, and these cuts actually can be then used with other teams for testing. We can use them for histology test. We can use them for test for decellularization. So basically, the first experiments for decellularization, they are on a petri dish. So they have this yeast as a petri dish or a plate. So 24 well plate. You can have plethora of these. It's going to place them in, and you can test different strategies. But it's essential to really have enough samples, because you are spending a lot of force just for analysis. And you need to calibrate the analysis and so on, so the consumption is actually quite interesting. So these are marked for as recellularization with the green dot. So these has been selected for testing the recellularization. And there is a multiple vials filled with these, usually by 3. So if you go inside, you can see there's a whole batch marked with green, again marked with green, and again marked with green. So these are samples that are going, there's 3 of them in each, so there's 9 samples that will be now further going into the recellularization experiments. Sure. So more and more are coming, and more are processed, decellularized, and then put into other experiments for analysis.
So is that a mix of 3D and 2D free cellular samples?
Yeah.
Okay.
These I can check it to 2D or 3D. 3D. So this was 3D, and 2 there's a 2D.
Thank you.
So the banks, but the outcome is the same. The only downside of using a precedent tested 2D, 3D differentiation, and also the dynamic and non-dynamic decellularization. The dynamic is in bioreactors, same water constant chemistry, and the non-dynamic or stationary, that's using the same chemistry but it's in a vial and it's shaped in precedent.
Okay, like a flask or yeah, holding vibe. Okay.
There's plus something like this that you can seal as a gasket and have to seal it inside, and then it's steadily sealed. Then you can
So that's your non-dynamic.
that's your non-dynamic protocol. Both of them works. The non-dynamic is really cool that you can passphrase because the virus is only one. So it's a really cool thing to work with. When it will be, you can do insane amount of experiments and we can really deep dive into literature. But if you want to mass produce, let's say, at the one moment you got like 9 or 10 ore skins, and you really need them because these guys need it, you will store something, and then will take a lot of time. It's almost 9 weeks. Impossible. We need to really adhere to the schedule. We really need to follow the timeline and really try to hit all the milestones. So basically, the outcome is the same, but static one can help us mass produce if we want to further.
Thank you.
Then if we go to the bioreactor room, follow me. As I mentioned, there are 2 of them. So first one, that's a decellularization setup, and behind me that's a vassalization setup, or some.
So can we see the second bioreactor?
Yep.
That's so if we had taken this over, we had taken out the decellularized matrix and we were getting ready to recellularize the whole matrix.
Yeah.
That would take us over to this
Rather
part.
just take us over here.